In previous lectures, I've discussed both photosynthesis and cellular respiration, but we haven't gone into much detail about either of them. The next two lectures are going to focus on these two topics. We're first going to talk about cellular respiration, and then we will have a lecture about photosynthesis. Both of these reactions have to do with energy and energy conversions. So we'll talk about how we take the products from photosynthesis and use them to power our cells in our bodies. And so we will go over all the different aspects of this chart in the next couple lectures. To put in perspective, you are going to use about a 1/4 pound of glucose each day in just your brain. Okay, a 1/4 pound. So you could weigh that out, get some sucrose. Sucrose is fine to look at instead of glucose, and weigh it, and a 1/4 pound of that is what you are actively burning through a day, and you're using the energy that's stored in the bonds of the glucose. So we require an enormous amount of energy in order to power our normal bodily functions. And today we're going to actually look at how the cells are going to harness the energy that is stuck inside the glucose. We're talking about energy throughout this entire lecture, but we need to know where it starts. So everything that is living requires energy to continue to be living. Almost all of the ecosystems on the planet are going to get their initial energy from the sun. So solar energy will be coming in, plants will convert that into chemical energy in the form of glucose, and then we can have energy through that process. So there's a transformation of energy from solar to chemical. But almost all of the ecosystems on the planet are going to use the sun as the initial source of energy. There are a few other types of energy sources that are not the sun. For example, there are some geothermal deep sea vents that also will have some energy that's associated with them. Now, during the process of photosynthesis, plants will take solar energy and will convert it into chemical energy in the form of sugar. They can also make other organic molecules. Now, they're going to do this by taking inorganic molecules like water and carbon dioxide and combining them to form things like glucose. Okay? Throughout an ecosystem, you will have movement of energy from producers, which are going to be the groups that are going to initially make the transformation of the energy from solar energy to chemical energy. And then we can have consumers, which are going to eat that energy and use it to power their bodies. Ultimately, every living thing will die, and all of the organic compounds that they're made up of will then be broken down by decomposers, which then will return all of those inorganic nutrients to the soil and to the environment so that they can be used for new life forms. Okay? So all animals, including us, will depend on photosynthesis in order to survive. Okay? We can either eat directly plants, or we can eat other organisms that eat plants. Now we're going to start talking about different types of organisms and how they get their energy. And there are two major forms. We have what we call autotrophs. The word autotroph means self-feeder. These are organisms that are going to be able to make their own energy on their own. They can take inorganic compounds like carbon dioxide and water, and they can bind those together to make organic molecules like glucose, lipids, carbohydrates, nucleic acids. Okay, so they're going to be able to make their own. So they can make their own from just inorganic, non-living matter. Okay, so they can get carbon dioxide from the air, they can get water, and they can get minerals from the soil. So different components they can get from the soil to help build all those different parts of those biological molecules that we need. The second category is heterotroph. Hetero means other, so these are other feeders. They cannot make their own organic matter. They have to eat autotrophs in order to get that organic matter into their bodies. Okay, so we cannot, as humans, go out and kind of lay out in the sun and take the sun energy and turn it into glucose, but plants can. And so we will always rely on plants and their ability to make those things like glucose. Okay, so heterotrophs will include all animals, including humans, and anything that is not going to be able to make their own food. Okay, so we cannot make our own organic molecules from inorganic. A different category that is similar to autotroph and heterotroph is this idea of what we call producers and consumers. So most ecosystems will rely on photosynthesis for food to be produced initially. Okay, so we have to have the energy that's stored in the sun, our sunlight energy, to be converted into chemical energy in the form of glucose and other biological molecules. Those can then be eaten. When we have the groups that are autotrophs that can produce their own food, we consider them producers. They're producing the energy. They're technically doing a conversion of energy. We don't create energy or destroy energy, but we can convert it. And so the producers are converting the energy from the sun into a usable chemical energy. So they produce food. Groups that are going to be producers are plants, algae, and phytoplankton. So all of these are going to use sunlight in order to produce their food. Heterotrophs are considered consumers. So think about humans as consumers. We consume the products that are made by the producer. We can eat plants. We can also eat animals that eat plants. Okay, but we're eating for all of our energy. We cannot make it on our own. We have to actually take in the food, consume it, and then break it down and extract energy that way. Animals will be in this group, fungi and zooplankton. So none of these groups are going to be able to kind of do something like photosynthesis. Okay, so we consume food by eating plants or by eating animals that eat plants. So a good example, not that anyone eats koalas, but people eat cow. So cows are going to be eating the grasses, so they're consuming that. We then eat a cow, and therefore we are eating plants indirectly by eating the cow. Now there's going to be a chemical cycling between photosynthesis and cellular respiration. We go back and forth with the different products. And so it's really interesting how these are tightly connected to each other. So the end products of photosynthesis will go into cellular respiration, and then the byproducts of cellular respiration will go into photosynthesis. Now the main two ingredients that we're going to start with for photosynthesis is carbon dioxide and water. So these are both inorganic molecules. We have carbon dioxide and we have water. And through the process of photosynthesis, those will be combined to form glucose. Okay, CO2 will be absorbed into the plant through very tiny pores that we'll talk about. They're called stomata. They're on the surface of the leaf, and CO2 from the air will go into those holes. And then water will be absorbed mainly by the roots from the soil. Okay. Inside of the leaf, we will have these specialized organelles called chloroplasts. And chloroplasts are going to be able to take sunlight energy, capture it, and then incorporate it in the bonds of glucose. And that will be what we talk about on our next lecture. We'll talk completely about that process of how all of that takes place. Now, the byproducts of photosynthesis are glucose, which is what we need to use to consume, but oxygen is also a byproduct. And you may think that's weird, it's kind of, it's not usable for the plant, but it's completely usable by humans because we are going to do respiration, and part of respiration is taking in O2. So those byproducts that are made during photosynthesis will then go straight into cellular respiration. So we will have glucose, which will be broken down, and the energy that's stored in the bonds of the glucose will be extracted. But we also will use oxygen. So oxygen is an absolutely necessary molecule for cellular respiration to take place. The whole point of cellular respiration is to make ATP, which is adenosine triphosphate, which is the actual energy currency that we can use in our bodies directly. So we can't use glucose directly to power different chemical processes and cell processes, we actually have to turn it into ATP in order for it to work directly. So both plants and animals will perform cellular respiration because they both will need ATP in order to power the different processes in the body, but you will only see chloroplasts and photosynthesis taking place in plants. Okay, so you do not see them in both plants and animals. So mitochondria, where we are going to have our cellular respiration take place, mitochondria will be found in plants and animals equally, but you will only find chloroplasts inside of the plants. Now, the waste products of cellular respiration are going to be carbon dioxide and water. So one of the things that we breathe out when we do our respiration in our bodies, we breathe out carbon dioxide, and that carbon dioxide is going to go directly into the photosynthesis process. Now, for a plant, they're performing photosynthesis to make organic molecules to help build the structural components of their bodies. So we learned about cellulose. Cellulose is going to be a bunch of glucose molecules that are strung together. Those glucose molecules are then going to form these fibers that give the plant's structure. But they also produce glucose for energy and fuel. If they produce too much glucose for kind of fuel and they don't need it for growth, they will actually store it in things like potatoes. So the potato, which is below ground, which is, right, we can see it in this picture, those are storage, sort of like storage bins for the plant. So they're going to store excess glucose in the form of starch in these, in this case, these are potatoes or tubers. And when the plant needs extra glucose, it can then extract the glucose from the starch in the potato and can use it. Now, because the plant makes these, we can then take advantage of it and eat the potato and get the starch from that. Okay, and so that's how we're going to start using plants in order to fuel our bodies. So we're now going to go more into depth about cellular respiration. And cellular respiration is called an aerobic harvest of food energy. This aerobic harvesting of chemical energy is going to require oxygen. So the word aerobic means requiring of oxygen. So if you look at this image with this woman who's running, and you've probably run or done some type of athletic event where you, or anything, because living requires this, but you breathe in oxygen. The oxygen is going to be taken into the lungs and then put into the bloodstream. The blood will then deliver the oxygen to all different cells in the body. This specifically is showing you muscle cells. So oxygen will be deposited at the muscle cell. The muscle cell will then perform cellular respiration, and one of the byproducts is going to be CO2. We then need to get rid of the CO2 because we cannot have too much of that in our body. So we breathe it out. And this is where the idea of this respiration. So if you think about human respiration, we tend to think of breathing, which is correct. When we talk about cellular respiration, it is using the product of breathing and using oxygen as a part of this larger extraction of energy from glucose. Okay, this is going to be the main way that we will harvest energy from the food that we eat. Okay, so we take the food that we eat, we break it down into its smaller components. We're mainly going to talk about glucose. So if we eat a potato and break it down to smaller pieces of starch, and then the starch can be broken down into glucose, that glucose will then go and be in the cell and be ready to go through this process called cellular respiration. Again, there is a requirement of exchange of the gases. Because that's happening, that's one of the byproducts and is used for cellular respiration. Okay, so cells will take in the oxygen and then they will get rid of CO2. We're now going to go into cellular respiration. I'm first going to give you an overview of the reaction that's taking place, and then we'll go into more detail. So this is the reaction that's going to take place for cellular respiration. We take glucose we're going to have oxygen, and then through a series of chemical reactions, we will then produce carbon dioxide and water. And the main thing that we're trying to make right here is ATP, so the energy. And we're going to make approximately 32 molecules of ATP for every 1 glucose molecule. Now, this process, this chemical reaction, is going to be extremely important. Okay? Almost all organisms are going to perform this process in order to get energy for the different things that they need to do. Okay? And so it's going to provide the energy for all the cells to maintain the functions of life. Okay? So I would definitely know the overall equation for this. I want to also point out that when we're looking at these reactions, you're going to realize that they're balanced, where you can see that the number of carbons, hydrogens, oxygens will be equal to the number of carbons, hydrogen, and oxygens that you have on the other side. So if you actually did the math out, you would see that there would be the same amount on each side. So we're not destroying anything, we're just rearranging these different bonds, and through that process we can extract energy. So here is the entire overview of the cellular respiration. So there are 3 main steps that we will talk about. The first step is going to be glycolysis. This takes place in the cytoplasm outside of the mitochondria. So this is the only step that's going to take outside of the out of the mitochondria. The second step is the citric acid cycle. This is the first step that's going to take place in the mitochondria. So you only have this taking place when you're inside. The third step is called the electron transport chain, and the electron transport chain is also going to be completed inside of the mitochondria. Now, up at the top of this image, you can see that both animals and plants will have mitochondria. So it's really important to remember that cellular respiration is a universal process in these animals and plants. It's not something you just find in animals, because plants also need to be able to power different chemical activities in their cells, in their body, in the structure. And so they're going to need ATP. So a little bit about glycolysis. Glycolysis is going to be a process where we split glucose into 2 molecules of something called pyruvic acid. So pyruvic acid is going to have carbons and oxygens, and it's going to basically just be the first step of breaking down glucose. So we're going to split the glucose molecule into 2 molecules of pyruvic acid. Again, it takes place in the cytoplasm, so you would not see this happening inside of the mitochondria. The citric acid cycle, which also can be called the Krebs cycle, I tend to call it citric acid cycle, not Krebs, but they are interchangeable. That is going to continue the breakdown of glucose, and it's going to break glucose down into CO2. So at the end of it, we're going to have the CO2 that we're going to release. Now, during this process, we are going to be extracting energy from those bonds that we're breaking, and we'll talk about how that's going to happen. Okay. CO2 is a waste product, so the body does not need this. It doesn't need it in plants, and it doesn't need it in animals for at least cellular respiration. So it's going to be breathed out in humans and other animals, and then in plants, they'll have smaller amounts, and then they actually could use it for use it for photosynthesis, but I don't think it's a very large amount, so they usually have to take more CO2 in from the environment. The citric acid cycle will take place inside of the mitochondria. The mitochondria is going to have specialized enzymes that are only located there, and those enzymes are going to be part of this process of citric acid cycle and the electron transport chain. Okay, so again, the reason why it's in the mitochondria is because that's where the enzymes are going to be. The whole process of cellular respiration is taking the energy stored in the bonds and convert it into ATP. Now, the way that this is going to take place is when we're doing the process of glycolysis and citric acid cycle, we're breaking the bonds. but the energy is not going to be directly turned into ATP, at least not much of it at that point. Okay, most of the energy that is stored in those bonds is going to be taken, stored for a very brief period of time, and taken to the electron transport chain. There is a little bit of energy in the form of ATP that is produced through glycolysis and citric acid cycle, but it's very, very small. You make about 2 molecules of ATP during glycolysis and about 2 molecules during the citric acid cycle. However, the majority of the energy will be produced when we do the electron transport chain. So in order to take the energy that's in the bonds of glucose and pyruvic acid that we're breaking down, we're actually going to transfer that energy in the form of electrons to a molecule called NAD+. Okay, and this is nicotinamide adenine dinucleotide. And you can see this molecule over here. This is AD, has ADP on it, has a ribose, and then it's going to have in this case, an adenine. Okay. So this molecule right here is going to be able to hold electrons, and I drew a little picture for you guys. So you can kind of think of it as a little energy shuttle, and it holds electrons in its structure. So it'll kind of grab onto those electrons, and it will then turn into NADH. Okay, so when it's NAD+, it doesn't have the electrons on it, but when it's NADH, it means it has electrons. And so these electrons will then be transmitted to the electron transport chain. Okay, and this is going to happen during glycolysis and the citric acid cycle. So we're going to make molecules of NADH that we're then going to take, and they're going to deposit those electrons at the electron transport chain. The third stage of cellular respiration is the electron transport chain. This is when we take all of those electrons that were captured on the NADH. They will then be moved to this electron transport chain, where we will then do this process. We combine them with oxygen to form water. Okay? During this process, other molecules will be moving, and we will then be able to produce energy. And so this is very abstract right now, and I'll show you how this works. But the overall product is that we're going to make a bunch of ATP through the transfer of these electrons. And the electrons are going to be passed through these molecules that are embedded within the membrane of the mitochondria. Okay. When the electron transport chain transports the electrons from NADH to oxygen, that's when we will then start releasing the energy in the form of ATP. Now, I'm going to take a brief aside before we go into the more details about each of those steps, because a lot of what's happening in these chemical reactions is something called a redox reaction. And a redox reaction is when you're transferring electrons from one substance to another. In a redox reaction, you have something that is being reduced and something that is being oxidized. And these two terms are going to refer to either gaining or losing electrons. When a molecule is oxidized, and in this case, glucose is going to be oxidized, it's going to lose electrons. Okay, so we've talked about electrons. They are going to be important in bonding. And so when electrons are actually stripped off of this and broken because of the bonds, they will then be oxidized. So glucose will be oxidized, which means that at the end, we're ultimately going to have something like CO2. So they lose electrons. Reduction is going to be the acceptance of electrons. So in our overall reaction, oxygen is going to be reduced because it's going to gain electrons in the form of hydrogens. Okay? And now you might say, why is it called reduced? Well, it's called reduced because if you think about it, if you gain electrons, you are having a higher negative charge. So you're reducing your charge, which means you're more negative. Okay, so oxidation is going to be losing electrons. We don't really call it, you're not becoming more positive necessarily in this situation, but we definitely call it reduction when the oxygen is going to then gain electrons in the form of hydrogens and form water. Okay, so it's going to reduce the positive charge. In cellular respiration, the main thing I want you to know is that glucose is oxidized, so it's going to lose electrons and hydrogens in this entire process. You can tell that it's losing electrons and hydrogens because you go from something like glucose that has, carbons, hydrogens, and oxygens to a molecule that only has oxygen and carbon. Okay. The oxygen is going to be reduced because it's going to gain electrons and hydrogens. So you're going to see the hydrogens show up onto the oxygen. Okay. So this is going to be how that redox is going to take place in cellular respiration. Now, this idea of removing the electrons and hydrogens from chemical bonds is going to release energy. And the reason why oxygen is necessary for cellular respiration is because oxygen is very electron grabby. It wants to grab electrons like no other molecule that we talk about. It's going to try to strip electrons off of other molecules so that it can fill in its outer shell. Okay. So there's nothing that we will talk about that attracts electrons stronger than an oxygen atom. So when the hydrogens and the electrons that are associated with those hydrogens move from glucose to oxygen, they are actually falling. And through this process of falling, because they're going to move from one molecule to another, they're going to release energy at every single one of those steps. Okay, so the potential energy stored in the bond of when we break that bond will be the release of energy. Okay. That electron that is then being released from that, in this case, glucose, is then going to be attracted to oxygen because, again, oxygen really wants to attach to those electrons. So when water is produced and we move the electrons from the glucose to the oxygen, that will have an energy release that accompanies it. Now, just to put this in a little perspective, if you do a redox reaction between hydrogen gas and oxygen, there's an actual explosion. So it's rearranging how the hydrogens and electrons are being bound to the oxygen because the oxygen is going to try to grab those. So if you have these two molecules, they will actually cause an explosion and then water will be released. So we know that there's a lot of energy that's stored within these bonds. Okay, and so that's the reason why we are doing this whole reaction in order to extract the energy that are on those bonds. Now, the reason why we do it stepwise, and we'll look at the electron transport chain where we're not kind of going directly from glucose to oxygen and transferring the hydrogens and electrons to water to form water, we're doing it stepwise because we do not want an explosive reaction to happen inside of our bodies. Okay, so we have to do it very slowly so that we can release the energy in a controlled way and extract that energy for the production of ATP. So here we have the overall equation again. Remember that we're taking glucose and water and through a series of reactions, so you can think of glycolysis, citric acid cycle, and the electron transport chain, we will produce carbon dioxide and water as well as ATP, which is our main goal. We want to make the ATP. So this equation shows that we are just rearranging all of the atoms of these molecules. We're not adding new stuff. We're simply taking what we have and arranging them, and in that rearrangement of where things are bound, we'll be able to release energy. Okay, and so this will then produce that ATP that we can use for cellular work. At the end of cellular respiration, we make approximately 32 ATP molecules. It is not always 32. It can sometimes be higher or it can be lower. And it depends on the cell and other things that are going on in that cell. So it's not always going to be a strict 32 ATP. That number can vary slightly. We're now going to go into detail about the different chemical reactions that are going to take place in the 3 different stages of cellular respiration. Our first stage is glycolysis, and I want to remind you that this is taking place in the cytoplasm. With glycolysis, we start with glucose, and in this case, we're going to have our glucose molecule written in a linear form. It has 6 carbons. Our output is going to be 2 molecules that are 3 carbons each, which are called pyruvic acid. You definitely want to know what your input is and what your output is. Now, another important step that happens during glycolysis is that we do require some investment energy. ATP is required to break the bond between the glucose. So we have to invest energy in order to gain energy at the end. So we invest 2 molecules of ATP, which is then going to allow for the breaking of the glucose molecule. Okay, that glucose molecule, we call this the investment state, will then allow for the production of 3 carbon molecules that will then donate their electrons to NAD+. So we have 2 NAD pluses right here. The electrons will be stripped from these 3 carbon molecules and will be placed on these NADHs, like I mentioned. So this is an electron, so they have little negative signs because they're electrons. This NADH is going to be taken and is going to be moved to the electron transport chain. So we'll come back to these. In all of this process, there are many steps that are taking place, but the main thing that's going to happen is that we first of all broke the glucose into 2 3 carbon molecules, and we also are going to extract some ATP. We extract 4 molecules of ATP, so one of the 3 carbon molecules will allow for the production of 2 ATP. The other one will also make 2 ATP. So we have a total of 4 ATP that are produced. However, the net amount of ATP that we make is only going to be 2 because we used 2 to start with. So at the end, we only really are making 2 more molecules of ATP. Okay, so we are finishing glycolysis with the output of pyruvic acid. Okay, these 2 pyruvic acids are then going to move on to the next step. Our next step is going to be the citric acid cycle. So we take the pyruvic acid that we just created, and we're only looking at one of these molecules, although we did make 2 from the previous step. So 1 molecule of pyruvic acid is going to come in and is going to want to enter into the citric acid cycle. However, pyruvic acid is not going to be able to be used directly. It needs to be altered slightly. So pyruvic acid is actually going to have a carbon stripped off, and we're going to lose it in the form of carbon dioxide. When this happens, pyruvic acid turns into something called acetic acid. The acetic acid is then going to donate some of those electrons to NADH. So we have another group of electrons that are going to be able to go down to the electron transport chain. And in this process, acetic acid is going to bind something called coenzyme A, which then is going to produce a molecule called CoA or acetyl-CoA. Acetyl-CoA is the output, and this will be able to enter the citric acid cycle directly. Okay, so we have to alter the chemical arrangement of pyruvic acid before we are allowed to enter into the citric acid cycle. Once we have acetyl-CoA will then move into that next step. Once we have acetyl-CoA, we can then go into the mitochondria, and the acetic acid will then enter into the citric acid cycle. The citric acid cycle, like its name implies, is a cycle. And so what's going to happen is we'll have acetic acid that will enter into the cycle. It'll actually join with a 4-carbon molecule to form something called citric acid. That will then go through a series of steps, and the whole point of this process is to break down this acetic acid into carbon dioxide. So you're going to produce more carbon dioxide during this process. And this acceptor molecule will just keep on going through this process. It will accept new acetic acids and then will kind of go through. What I'm not showing you is that there are a series of different events that are taking place in the citric acid cycle. It is quite complex. There are many enzymes that are involved, and the arrangement of the molecule is quite intense. So there's lots of rearrangement that takes place. We will not focus on that for this class. Our main focus is on, what is input and what is output. So our input is going to be that acetic acid. Okay, we also are going to have some of these, another molecule. We have NADH, our NAD+, that's going to be able to take a couple electrons. We also have now a new one called FAD, which is very similar to NAD+. It also will hold some electrons. So while we're rearranging things, we're going to have electrons that will bind to the NADH to form a NADH and FADH2. So these are both the charged form. That means that they have electrons. We're also going to produce a little bit of ATP, and we're going to have an output of carbon dioxide. Now at this point, carbon dioxide is a byproduct and is waste. We're going to just get rid of it. So at this point, we've extracted all of the electrons from it that we want. And we are now ready to move into the next step, which is going to be the electron transport chain. So we're going to take all of those electrons that we've stripped off of the glucose molecule and put them into this electron transport chain. The last stage of cellular respiration is going to be the electron transport chain. Now what we see here is the electron transport carriers that are actually embedded within the inner membrane of the mitochondria. And you're going to notice that there's proteins that are embedded within this membrane. And their function is going to be that they're going to move hydrogens out into this intermembrane space. So they're going to pump hydrogens through the membrane to this other side. Now, the main point of the electron transport chain is that we are going to take electrons and we're going to pass them from these electron carriers that are the proteins that are embedded within the membrane. And so the electrons are going to be deposited to these electron carriers and be moving from one carrier to the next until they finally get to oxygen, where they will be added to oxygen and we will get H2O. Now, as the electrons move, they are allowing for energy to be released, and we can pump these hydrogen ions against their concentration gradient. Okay. Now, in order to understand this, I want to kind of use an analogy. So let's look at the electron transport chain in a kind of different format. So we always are going to start with the glucose. So we have this man down here with the glucose, and the glucose is going to provide the initial energy. So it has all the energy stored in its bonds. The electrons from the glucose are going to be added to a molecule called NADH. So this woman is going to kind of be the symbol of the NADH. So she's going to grab on to this electron, and then she's going to pass it off to this electron transport chain, and the electrons are going to move through the chain, and while they do that, they're going to actually turn this wheel, and this wheel is going to help produce ATP. So in the cell, it's going to look a little more like this without people. We're going to have the NAD+, which does not hold any electrons. Electrons are going to be stripped off of the glucose and added to the NADH. Those electrons are then going to be delivered to the electron transport chain. When they do that, they're dropped off, and the electron transport chain is going to pass these electrons from each of those electron carriers in the membrane. As they do this, they are going to be creating ATP. The final electron acceptor is going to be oxygen, and the way that this is going to happen is the electrons, along with hydrogen, are going to be added to the oxygen, and we form H2O. So that is our byproduct of this. It's a waste product. We don't necessarily need it. And so it's going to be kind of the final location where those electrons go to. But as we've done this, we're moving them down, and as we move them down this electron transport chain, energy is being released. So let's look at this a little closer. Now, it's important to understand the mitochondria. The mitochondria structure is that it has kind of these outer membrane and then the inner membrane, and there are these invaginations that are going to kind of be this inner membrane space. This is a cartoon, and this is what a real mitochondria would look like. Now, in the membrane, we're going to have the electron transport chain, so it's going to be present within this membrane. Now, the membranes are highly folded. That increases surface area. So there's a lot of surface area in there, which means that we can have lots of electron transport chains. So we can do this process at a very high rate because we have so many kind of electron transport chains built in. So you can make sure that you're making enough ATP. Okay. The energy is going to be stored by the electron transport chain, and the way that they're going to do this is kind of like building a dam with water. So the water is going to be on one side of the dam, and then if we open like a hole in the dam, the water can actually flow through. And so I'm going to show you what I mean by that. So I'm coming back to this image. Again, I want to kind of get you oriented to where we are. So we have kind of a zoom in right here. So we are the mitochondrial membrane. So this is the inner membrane. We're going to have kind of inside the inner membrane and then the space outside of the membrane. And what's going to happen is instead of water being pushed, one direction, we're going to pump hydrogen ions against their concentration gradient into this smaller space of this inner membrane. This is going to build up a dam of hydrogen ions, and we then are going to have a pathway for the hydrogen ions to rush back into this inner membrane. And while they do that, they're moving down their concentration gradient, so they will naturally move down this direction. When they go through, they are going to go through this structure called ATP synthase. And ATP synthase is going to produce ATP. You can think of it as kind of a turbine. And so it's going to, through letting the hydrogen ions into this membrane, it's going to allow for the production of ATP. Okay. Now, how are we pumping these ions? If you remember before, I said that electrons, when they're passed from one electron carrier to the next, they're releasing energy, and so hydrogen ions can be pumped against their concentration gradient. Okay, and so they'll move down from one to another. We have FADH2 donating electrons at this point in the electron transport chain, and they'll keep on moving down. Now, what you're going to end up with is water and ATP. Okay, CO2 was made in the earlier stage in the citric acid cycle. So this is going to be our byproducts, so the H2O and the water. Sorry, and the ATP. Now I want you to see this little skull and crossbones, and you might be like, why is there a skull and crossbones right there? Well, it turns out that cyanide, which is a very deadly poison, will actually bind to this electron transport or electron carrier in this electron transport chain and make it so that it cannot pass electrons. Okay, and so what happens is that you're basically disabling the electron transport chain and you cannot make ATP. If you're not being able to make ATP, you will eventually die. So cyanide will irreversibly bind to this specific part of the electron transport chain, stop it from working, which means that you can no longer do this part, which means you can't do ATP synthase. So basically it's going to stop that and it'll kill you. Here's a GIF showing how this is going to work. So we pump the electrons to the intermembrane space. The way that they're being pumped is by the passing of those electrons. Okay, let's see if it goes again. I don't know if it's going to do that. So here we go. So hopefully it'll go to the next stage. All right, here we go again. So we have NADH is going to drop off those electrons. The electrons are the little black things that are going to move from one thing to another. They're pumping the hydrogen ions, and then the hydrogen ions are just going to flow right back in. There's no energy required. They actually produce energy in this process. Okay, so this is how the electron transport chain works. So here's the overall results. If we go back to glycolysis, we can make 2 ATP through direct synthesis. Remember that we do require a little bit of ATP to get this started, but we have a net of 2 ATP that's produced. The citric acid cycle will also produce about 2 ATP. And then when we get to this electron transport chain and the ATP synthase, it will produce about 28 molecules of ATP with a kind of average of 32 ATP molecules for 1 molecule of glucose. Okay. Now, I told you before, this number can change depending on certain situations, so it's not necessary to kind of know this exact number as being the firm always making 32 ATP molecules. Okay, so when we eat food, we are not going to eat just carbohydrates, and so we have to talk about other food molecules. I was talking mainly about glucose because glucose does go directly into the pathway, but you actually can have other food molecules enter into the pathway at different stages of this process. So there are different metabolic routes for carbohydrates, fats, proteins, and you can see, so here's foods. We have, looks like some type of sushi. I'm not sure exactly if there's like a California roll, but it has salmon on the outside. But we're going to have some vegetable and fruit. It looks like this is a cucumber, which is technically a fruit. And then I can't really tell what else is in here, maybe crab. Then we have the rice, we have seaweed, and we also have the salmon on the outside. So lots of different food types. So proteins, fats, and carbohydrates are going to be a part of this. So carbohydrates are going to enter through glycolysis. So that's where we would turn our carbohydrates into our glucose molecules. But we also can come through the fats. So fats can be turned into glycerol, and then or fatty acids, and fatty acids can go into the acetyl-CoA portion, or glycerol can go in through glycolysis. And then the proteins can go basically in at all the different stages. So there's amino acids that are needed at each of these different steps, and that's how we're going to be using them and making ATP. Okay, so there is a lot of interactions between all of these pathways. The main one we focus on is glucose. So glucose entering and going through glycolysis, and then citric acid cycle, and then finally electron transport chain. Now, there are situations where there isn't enough oxygen. So we talked about cellular respiration as being an aerobic situation. But there are times where we're going to have something called fermentation take place. And this is an anaerobic process. Anaerobic meaning no oxygen is necessary. And you've probably heard of fermentation mainly when it has to do with food. So we ferment a lot of different food products. So soybeans, we can make tofu, we can make soy sauce, we can make alcohol, we can make bread. All these things are going to deal with fermentation. So we're going to talk a little bit about this process. Okay, so all of this is going to happen without oxygen. We as humans can do it for short periods of time, but we mainly are going to do aerobic respiration. Okay, anaerobic, so when you have the letter A or the letter AN in front of a word, it means without. So this means without oxygen. And when it's without oxygen, we call it fermentation. It's no longer cellular respiration because you don't have the respiration part of it. Okay. It's going to rely only on the very first step, which is glycolysis. So if you go back and remember glycolysis, there's no oxygen that's going to be important in that step. There is an important part of both citric acid cycle and of the next step, which is the electron transport chain, that does require oxygen. So you do need oxygen at that step. But you can do glycolysis all by itself without any oxygen. So glycolysis doesn't require oxygen. It will produce 2 ATP molecules, but we're going to have kind of a different pathway after we make the pyruvic acid. Okay, so glucose can be broken into the pyruvic acid, but we'll have a different step after that if there's no oxygen present. So this is what fermentation looks like in human muscle cells. So you have the same process of your input being glucose. You then are going to break it down into 2 molecules of pyruvic acid. It does require a little bit of ATP to get started, but we do have a net of 2 ATP being created. Now the problem with this is if there's no oxygen, the pyruvic acid cannot go into the citric acid cycle. And so the pyruvic acid instead gets turned into another molecule called lactic acid. Okay, now the lactic acid is something that you probably have heard of. It is something that a lot of people talk about when they're working out and when the lactic acid builds up and it makes your muscles burn. And so that is going to be what happens in human cells. And we'll talk a little bit about this idea of the lactic acid. But the main picture of this is that this happens when there's no oxygen present. So let's talk a little bit about why muscles burn when we exercise. So this is something that was investigated by a biologist named A.V. Hill, and ultimately he will win the Nobel Prize for his investigation of muscle contraction. So that's a little bit out of what we're going to talk about right now. We're going to mainly talk about the lactic acid, but he did investigate kind of why your muscles will burn. And he was one of the founders of exercise science. So if you're in kinesiology, this is someone that kind of started that field. Now, the way that he did this experiment is he took muscles and put them into a kind of a fluid that either prevented lactic acid from diffusing out of the muscle or a solution that allowed for lactic acid to move out of the muscle. Now, this was a frog muscle, and they stimulated the muscle with an electrical current. That's how muscles are going to be contracting in our bodies. Obviously, this is a little different because we're using a battery, but it's still the same process, so the muscles are going to contract normally. There is no oxygen in this system because these muscles are taken out of the frog, and so therefore there's no breathing taking place. And so what's happening is this muscle is contracting continuously, and the one that's in this kind of pinkish peach fluid cannot have the lactic acid leave the muscle cell. It just stays inside the muscle, so it's building up the lactic acid in this muscle. This muscle does not have a buildup of lactic acid because the lactic acid can move out of the muscle cells. Now what happened is that when they did this, the solution that allowed lactic acid to leave the cell allowed for this muscle to work for twice as long as this other muscle. So at some point, these muscles will fatigue to the point that they will no longer be able to contract. That has a more complicated explanation, but we won't worry too much about that. But the main finding of this is that if you have no lactic acid buildup in your muscle will last for longer. Therefore, Hill kind of came to the conclusion that lactic acid is what causes muscle fatigue. Okay. So the main result of Hill's work is that the lactic acid is causing muscles to fatigue and therefore burn. However, from the time that he came out with that kind of conclusion, other scientists have done similar experiments and they've had some contradictory evidence appear. So remember we talked about science and the fact that you have to be able to repeat these experiments, and if the information that you get from these new experiments does not correspond with what you found previously, you do need to change your conclusions. Therefore, many scientists believe that there were some issues with the apparatus and the experiment that he ran. One of the main problems with Hill's demonstration is that he did this experiment at a temperature that is not the human body temperature. So our muscles are going to be performing at 98.6 degrees Fahrenheit, and he did his experiment at a different temperature, and so therefore you can't necessarily extrapolate the information that we learned from the frog muscles to humans. So we don't think that actually is going to work the same way in humans. We also know that certain individuals are unable to accumulate lactic acid, and their muscles fatigue more rapidly, which is the opposite of what you would expect. So you think if someone is not able to accumulate lactic acid, then they would have muscles that never fatigue, but it's actually the opposite, which completely contradicts what Hill initially found. So it seems that one of the reasons why muscles fatigue and start to burn is because of increased levels of other ions and not really lactic acid. But it still is controversial and we don't really know. Okay, so it is something that people are still looking into because people are very interested in how to resist muscle fatigue when they compete in different athletic competitions. Now, moving away from humans, other organisms are also going to be able to do fermentation. We're specifically going to talk about microorganisms. So yeast is a eukaryotic organism. It is single-celled. It is a fungus. And yeast is probably something that you're familiar with. We make bread with it. It can also cause different types of kind of irritations on our skin. So you can get yeast infections or athlete's foot and things like that. Those are kind of yeast-based. So yeast generally will do cellular respiration the entire process, but you can put yeast in an anaerobic environment and they can survive by doing just the glycolysis part and do fermentation. Now, the waste products of certain microorganisms are different from what we make as humans. So we make lactic acid as a byproduct, but other organisms are going to make something called ethyl alcohol. So ethyl alcohol is going to be what our alcohol that we drink is made out of. Okay? So here we have the whole process of fermentation in yeast. So we have glucose that comes in. They're then going to do the same process where we're breaking up the glucose into 2 molecules of pyruvic acid, but it can't go anywhere because there's no oxygen. And therefore, in this case, the pyruvic acid is going to be converted into ethyl alcohol. So this ethyl alcohol will then be what we can then use for alcohol. Here are just a couple different food products that we make with microorganisms and fermentation. Okay, so this fermentation is going to be important in when you do, sourdough, when you make kimchi, soy sauce, alcohol, and cheeses. So we're allowing these microorganisms to kind of do fermentation, and then we can use the byproducts. Here's a chart of some of the different fermented products that we use. So pyruvic acid is always going to be our starting point, but it can then be converted by different organisms into different products. So for example, we have, I don't even want to know how to say this first name, but it makes carbon dioxide and this propionic acid. We have other microorganisms that make lactic acid, which are important in like cheddar cheese and soy sauce and yogurt. Saccharomyces, which is a type of yeast, is going to produce ethanol. So that's where our beer and our wine is going to be from. We have other ones that are going to produce things that we wouldn't want to drink, like acetone and isopropanol. And then these other microorganisms right here are going to make acetic acid, which can be found in vinegar. Okay, so this whole process is going to happen because of fermentation, and then we can use the byproducts of that fermentation for ourselves. The last thing I'm going to discuss in this lecture is this idea of the importance of oxygen. If we go back to the very beginning of the Earth, when it was formed, there was little oxygen on the planet. And therefore, the earliest life forms were not being able to do cellular respiration, but we do believe they were doing glycolysis. So glycolysis is a universal energy harvesting process. So if you look at all different types of life forms, they're all going to be able to do this process of glycolysis. If we look at the evolutionary stages of how this could take place, we know that between 3.5 and 2.7 billion years ago, there was not a lot of oxygen on the planet. And therefore, the earliest life forms, which we call prokaryotes, they were probably using glycolysis exclusively to make their ATP. Okay, it wasn't until oxygen started being at higher levels that the later stages of cellular respiration could start to take place. So here is a timeline of when oxygen was on the planet and then when life forms appeared. So we believe that the origin of the Earth is about 4.3 billion years ago, or maybe even, that looks like more than 4.5, so maybe 4.6. And we don't see our first fossils of prokaryotes until about 3.5 billion years ago. There's most likely going to be older forms of life, but we don't have actual fossil evidence of that. During this period of time where we have this blue bar, we're going to have very, very little oxygen on the planet. The very first oxygen is going to appear closer, into the 2.8 billion years ago time. It wasn't until about 2.2 billion years ago that the level of oxygen reached 10%, which is a modern day level of oxygen. This is when we think there then would be the evolution of these other stages, the citric acid cycle and the electron transport chain would start to evolve. It's not until 2.1 billion years ago that we have our very first eukaryotic organisms showing up. Okay, and then this would be what we have today. So this oxygen level is going to be really important. When we talk about the first life forms on the planet, we know that the presence of or the absence of oxygen was actually essential for life to form. Oxygen is a very corrosive molecule, and I told you before, it wants to grab onto electrons, and so it can be really disruptive with chemical bonds. And so we think it was really important that there was no oxygen on the planet at the very beginning in order for life to begin. It wasn't until later that there started to be other life forms that were able to deal with the oxygen. Okay, and we will mention kind of how this process took place in the very first early life forms in a couple lectures. Okay. So glycolysis occurs in almost all organisms, which suggests that it was kind of evolved very early in the life forms, because if the common ancestor of all of us was able to do glycolysis, then that means that every living thing that is going to be that descendant is also going to be able to do glycolysis. So we can find in each of the domains of life, and we'll talk about the different domains, the ability to perform glycolysis. So it's going to be very important. Okay. We also know that the location of glycolysis is going to show that it was there prior to kind of eukaryotic cells. Remember that the glycolysis takes place in the cytoplasm. The rest of the process takes place in the mitochondria. But our very first organisms on the planet, things like bacteria, they do not have mitochondria, and therefore they're going to have to be able to do glycolysis just in the cytoplasm. It's not until later, when we have membrane enclosed organelles, that these other steps of cellular respiration can actually develop. Okay, so we are going to end right there. We will talk about photosynthesis on our next lecture. I want to just let you know I'm aware that this is a difficult topic. We will go over cellular respiration on our review session and kind of go over the main points we'll do it a couple times so that it gets really stuck in your head, because I am aware that this is complicated. There's a lot of steps, it's a lot of chemistry, but we just want to know big picture ideas. Okay, thank you.